In this video, we will discuss about the specificity of a CRISPR-HiFi Cas9 system against KRAS mutant. So, let us first understand about why the focus is on KRAS mutant. So, KRAS mutant is found to be in the present in many cancer cells such as lung cancer, pancreatic cancer, and it is one of the main mutant that makes the prognosis or diagnosis of the tumors difficult. So, the researchers wanted to make a system that can kill the mutant KRAS in the cancer cells, but at the same time there is minimum damage happens to the wild type KRAS that are present in the healthy cells. This is their idea. They wanted to develop a system that can uh, target this KRAS mutant but not the KRAS wild type. So their idea is shown in the panel B, where we can see that in the non-tumor where KRAS wild type is present, the HiFi Cas9 system does not target that KRAS, whereas in the tumor cells it targets the mutant, whereas in the tumor cells where the KRAS mutant is present, so their HiFi Cas9 system can target that mutant. And the sequence that they have shown it here. So basically they have designed a system where they have checked it for two different kind of mutations that are commonly found, that is G12C, KRAS G12C and KRAS G12D, where G is mutated with C at 12th position and G is mutated with D at 12th positions, and in the wild type there is no mutations. So they have tested different guide RNAs, and they have tested guide RNAs using two different PEM sequence. First one is AGG and second one is TGG. And when they check these two systems in the different cell lines which contains KRAS wild type and KRAS mutant, both the KRAS G12C and KRAS G12D, and what they are able to see is that when the results, the top row for the panel D shows the results for the G12C mutations for this P1 and P2, and they are able to see that both this P1 and P2 can edit this KRAS G12C. We can see the multiple bands, but there is no multiple bands shown in this KRAS wild type for both the cases. And the efficiencies for the P1 guide RNAs is high, so they have selected this P1 guide RNAs for G12C for the subsequent studies. And when they check it for the G12D KRAS mutations, they can see that for the case of P1, in the wild type case also they have seen the multiple bands, but and the efficiencies are high for the mutant. But when they use the P2 guide RNAs, they can see minimum multiple bands in the wild type KRAS and more multiple bands in the case of KRAS G12D, and here the efficiencies is high, also high. So they have selected for the KRAS G12C they will use this P1 guide RNAs, and for KRAS G12D they will use this P2 guide RNAs. And thereafter what it is, they, in order to further confirm the specificity, what it is, they mutate the sequence that is highlighted in the blue to, that is from G to C in both the guide RNAs, that is P1 changes to P1M and P2 changes to P2M, and then they check whether it will still cause editing or not. So when they use P1M, P2M and P1 in the wild type, they do not see any editing, but when they use in the KRAS G12C in the P1 case, they are able to see editing as they are expected in the similar to the results from the panel D. And for the P1M, P2M, they do not see any edited, but when they add this C into the cell lines, this guide RNA sequence into the cell lines, they are able to see editing, indicating that their guide RNA that they have selected is able to do specific editings. Thereafter, they further confirm the results using NGS sequencing. Therefore, they further confirm the results using NGS sequencing, and what they are able to see is that in the case of this guide RNA G12C, the reads for the mutants, that is G12C, is high, whereas the guide RNAs does not did any editings in the wild type, so the reads for them is minimum, and this is reproducible using multiple runs. And similar results they have seen for guide RNAs G12D also when they check the mutants. When they read the NGS sequence of the this cells that are treated with guide RNA G12D, they in the wild type case they do not find too many reads, but in the case of these mutant KRAS cell lines, they are able to see reads, and this is also highly consistent. So this NGS results further substantiate their findings that the guide RNAs that they have find out using this HiFi Cas9 system can target the mutant KRAS, but there is a minimum non-specific editing done on the KRAS wild type. So this is what they have finally concluded at the end of this figure. If you need to understand more about this experiments in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.